Welcome to Bioratna. I am Chandana Sengupta and today we are going to discuss the topic DNA versus RNA from the chapter Molecular Basis of Inheritance. DNA versus RNA. What we are going to see is that which one out of these two, that is DNA and RNA, which one is better genetic material and which one is better for the transmission of genetic information, better for transmission of genetic information. So we are discussing this for two molecules, that is DNA and RNA. But earlier it was protein. Protein was the preferred molecule and there was much debate that it is protein. Even it was preferred above DNA. It was thought that protein is the genetic material. Then it was Hershey and Chase. It was the experiment by Hershey and Chase which conclusively proved that no, proteins are not the genetic material and DNA is the genetic material. But of course there are some organisms in which RNA also acts as genetic material, like tobacco mosaic virus, or QB bacteriophage. This definitely means that RNA can also act as genetic material. DNA is acting as, in most of the organisms we see that DNA is the genetic material. But RNA is also acting as genetic material in case of some organisms. Now let us discuss why it is so that though both of them can act as genetic material, but DNA is found in abundance in most of the organisms. It is DNA which is the genetic material and RNA it is used for as a messenger molecule or as adapter molecule. Why so? Let us see. To come to any conclusion, we should first know, we need to know the criteria which a molecule should fulfill to become a genetic material. कोई तो क्राइटेरिया होना चाहिए ना जिसके बेसिस पे हम दोनों को परखेंगे, फिर दोनों को मार्क्स देंगे. So what are these criteria? The first criteria is that it should be, the molecule should be able to replicate itself. It should be able to replicate itself. उसे अपना कॉपी बनाने की पावर उसमें होनी चाहिए. Now both DNA and RNA, they can form their replica due to the rule of base pairing and complementarity. Both can form their replica. So let us give full marks to both DNA and RNA for this point, that they should be able to replicate themselves. Whereas proteins, the proteins, they do not fulfill this first criteria itself. So proteins, they cannot replicate itself. Of course, we already know by the brilliant experiment of Hershey and Chase that proteins are not the genetic material. But just to mention that the proteins, they are not able to replicate themselves. Proteins are formed from the instruction of DNA and the message carried out by RNA. That is a wonderful story which we are going to study in transcription and translation. So for this point, we have given equal marks to DNA and RNA. Let us move to the next criteria. The second criteria is that the molecule, to be a genetic material, the molecule should be stable. It should be stable chemically and structurally. It should be a stable molecule. Now, that means it should not change itself. It should not change itself either due to age or due to change in physiology of the organism or due in different stages of life cycle. Different stages of life cycle. कोई भी समय हो उसे खुद की स्टेबिलिटी को बरकरार रखना है. Then only it is fit for to be a genetic material. You have studied Griffith's experiment, Griffith's transforming principle. There you have seen that though the bacteria it dies due to heat, but its genetic material, it at least retains some of the properties by which it can transform the other bacteria also. So which one is more stable, DNA or RNA, and why? Let us answer this why. So which one is more stable? And we need to see that why this stability to more stability for one molecule. The first point, let me draw the diagram of the pentose sugar for both DNA as well as RNA. If this is for RNA, then and this is for DNA, then this is ribose sugar and this is deoxyribose sugar. Deoxy, oxygen is missing at one place. Which place? This position is C1. This position is C2. At C2 position, this ribose sugar, it has OH. This OH is very reactive and that's why RNA is very unstable. Come to DNA. This is, when we have written DNA, it seems that this molecule is DNA. This is actually deoxyribose sugar. This is deoxyribose sugar present in DNA. And this one is ribose sugar. This is ribose sugar. Fine. So here, this is C1, this is C2. At C2 position, only H is present, not OH, which is not at all reactive and it gives stability to DNA. Now, second point. In the nucleotides of DNA, thymine is present. Thymine is present, whereas in case of RNA nucleotides, they have uracil. This thymine, it provides lots of stability. This provides stability. That's why DNA is more stable. And the third point is that RNA has catalytic property. RNA has catalytic property also. DNA does not. RNA has catalytic property and DNA does not have catalytic property. Fine. So we have discussed 3 points which makes DNA more stable. What are they? This was the first, that is presence of OH group in RNA at C2 position of the sugar, making it unstable, very reactive, whereas DNA does not have the OH group. Second is this RNA, it has uracil in its nucleotides, whereas DNA has thymine. Thymine provides stability. And third is this RNA has catalytic property, making it more unstable. So here undoubtedly DNA is much ahead, and we give full marks to DNA for this criteria. It is very stable. Now let us see the next criteria. The third criteria is that it should be able to mutate. Ability to mutate. Why? Why the ability to mutate? So as to provide the scope for evolution. So as to provide scope for evolution. All of us want to evolve. एक ही जगह पे तो कोई नहीं रुकना चाहता ना. We are continuously evolving, and this, if our genetic material is not providing us the scope to evolve, then how can we evolve? That's why this genetic material, one of the criteria is that though it is providing stability, it should have the ability to mutate. One important point is slowly. So let us judge DNA and RNA for this criteria. Now RNA is mutates very fast. RNA is reactive also. RNA is reactive, RNA is unstable, and RNA is single stranded. Due to all of this, RNA, it mutates much faster. This is the reason why the viruses which have RNA genome and which have short lifespan, two criteria na? The viruses which have RNA genome and they have short lifespan. Short lifespan, so generation the gene will go on passing to the offsprings, and they are having RNA genome which is mutating very fast. So that offspring mutates very fast and changes into something different. So these viruses having RNA genome and short lifespan, they mutate very fast. This is the problem when so that we cannot prepare vaccine against them. Whereas DNA, it is providing stability. It is providing stability, but at the same time, it is providing us, it has the ability to mutate slowly. Ability to mutate slowly. That's why there is scope for evolution also. So here too DNA is the better material. Another very important criteria is it should be able to express itself. It should be able to express itself in the form of Mendelian characters. Mendelian characters. So first let us take DNA. For expression means DNA is dependent on mRNA, and then according to this information in mRNA, the proteins are formed. Proteins are enzymes. They are responsible for the different types of traits. That is the expression. So DNA is dependent on RNA for expression, whereas RNA, it can express itself directly. Actually, the whole machinery of the protein synthesis revolves around RNA, mRNA, tRNA, rRNA. So RNA, it is directly expressing itself. DNA is dependent on RNA for its expression. There is one middleman in between. DNA does not have any problem with that, and it can express itself through mRNA. Thus, we have discussed 4 criteria, students. During discussion, it seems long, But in your sticky notes, what you will do, you will write these 4 criteria in a single note. Fine. That is, it should have the ability to mutate. Short words simply. It should have the ability to replicate. It should be stable, and it should be able to express itself in the form of Mendelian characters. ये 4 ऐसे लिख लोगे. फिर दूसरे स्टिकी नोट्स पे जैसे why it is so stable, DNA why stable, why RNA is so unstable, तो उसके 3 पॉइंट्स आपने एक नोट में लिख लिए. Do it like that. Just look at these 4 slips and the whole thing will be clear to you. Now let us come to any conclusion. Mutate. Both mutate. Both can mutate. Both give us the opportunity to evolve. But DNA gives us the opportunity to evolve slowly. It is a stable molecule, but it provides us the ability to evolve. Whereas RNA, it mutates very fast. It mutates very fast. In one generation also it mutates. So here DNA is, we give marks to DNA. Next is replicate. It should replicate. Both can replicate equally. Both can replicate. So for this we give full marks to both. So full marks to, we give here full marks to both. But in case of stability, DNA is much ahead. So here DNA is much ahead. Fine. And express. RNA can express directly. DNA expresses through RNA, but it doesn't make any difference. RNA is always present there to obey all the orders by the DNA. Thus, the most important factor due to this stability character and the other which we have seen, so what conclusion we come to is that DNA is better molecule for storage of genetic information. Storage of genetic information. Whereas RNA is better for what? The transmission of genetic information, and together they are carrying out the work very beautifully. Hope you have understood the topic properly. Prepare the sticky notes. It will be easy for you. If you are new to my channel and if the topic is clear to you, then do subscribe, like and hit the bell icon so that you receive all the notifications. Thank you for watching the video.